We made a little facetious joke, Heart 2.0 beta, right? Because, I'm at Google, and I used to have Gmail beta for a long time until I got regular Gmail. And so I wanted to kind of refer to this idea that we're trying to do grow heart in a new way. And so first what I'm going to do is I'm going to give you a rationale for what is cardiac tissue engineering. Why do we even want to grow heart in the lab at all? And then give you some background on the heart itself. And then because the idea is that in order to grow heart really well in the lab, we need to know more about the native heart. Okay, so we'll start with the rationale for why we want to grow heart in the lab in the first place. Then, kind of go back and revisit the heart itself. And then I'll give you some selected and biased strategies for cardiac tissue engineering, because I'll really be going through my stuff. So it's a very biased, selected strategies for cardiac tissue engineering. But please feel free to stop me as we go along. It makes it much more fun for me and hopefully more informative for you. All right. So it's always nice to start with some artwork. This is the Fountain of Youth. This is sort of our inspiration for the field of tissue engineering. I mean, you see all the people kind of coming in on the left, hunched over and not doing so well, getting into this beautiful pool and then getting out on the other side, rejuvenated. And the kind of inspiration for the field of tissue engineering is kind of to search for this proverbial fountain of youth by being able to grow spare parts for the human body to help us extend our lives. And so, the idea is that, at some point, if we actually, are successful, that we will be able to kind of have a one-stop shop for organs. So transplants made to order, so to speak. So hearts and bones and cartilage and all kinds of things, as they wear out, we can replace them. This is the idea. So the motivation for the heart in particular is that, heart disease is such a big issue. I mean, we all know the statistics. Heart disease kills more people than all cancer combined, and as the world globalizes and the population ages, the toll is only going to continue to grow. And if you combine that with the heart's limited ability to regenerate and the lack of organ donors, we really have a strong rationale for being able to, for needing to find new solutions. In addition, we have a very limited ability to test new drugs. Imagine every time you need to test a new drug, you need to have a clinical trial, and so that there's a limited number of people with whom you can do that as well. And so if you combine all those things with just a basic desire to study cardiac development, we've got a really strong rationale for wanting to be able to grow pieces of human heart without, or get pieces of human heart that don't involve using a whole human being. Does that make sense? It's gotten some popular press. Recently, Wired had a cover story, much sexier looking than what my work is, on tissue engineering, and then of course it's been in the popular news press, as well as on, I don't know if anyone recognizes Grey's Anatomy, a show that I watch where they actually tissue engineered some cartilage. So this is a topic that's getting more and more in the public mind. So how do we actually grow tissue? Does anyone in the audience have any experience in working in a bio lab or having grown cells ever? Did I see a hand? No. Okay. So let me tell you a little bit about what it's like to grow tissue. So basically, we have you take cells out of their native environment, and we put them into what we call petri dishes. And if we keep them sterile, keep them moist, and give them food, we can grow them in incubators in these dishes quite well. We grow them, on the bottom of the dish, and you can look at them under the microscope, and they're growing. And this is kind of the traditional mode of cell culture for a long time, maybe almost 100 years. The problem is that in order to get these cells that we grow in a dish to form more complex tissues, it's much more of a challenge, because the petri dish isn't very much like the human body, and so the cells are kind of homesick for their native environment. The petri dish doesn't feel like what it would feel like. So for a heart cell, for example, the petri dish doesn't feel like what it would feel for that cell inside the heart. And so the cell doesn't respond the way it would, and it certainly can't build a piece of tissue. And so we need to do better to copy their natural environment in order to get them to not just survive, but to thrive. And so this is the rationale behind the tissue engineering paradigm, which doesn't use petri dishes at all, but in fact uses biomaterial scaffolds, so three-dimensional pieces of tissue. So the big difference is really that instead of growing cells on the bottom of the dish, we're growing cells in a 3D environment, so three-dimensional. And so in order to be able to do that, we have these biodegradable scaffolds on which we add cells and give them a lot of schmutz that they need, which basically means growth factors, sugars, proteins, the things that these cells need to really grow, and mix those into what we call cell culture media. This is like the food. It kind of mimics, there's some serum in there, it kind of mimics the nutrients that would be provided by the blood. And so the idea that if we culture cells in this kind of environment, that we'll do much better at growing 3D tissues. But what our lab does that's really, we got a hand up? Yeah.
Yeah, so it sounds like you're forming the shape with the scaffold.
Yes, we're forming the shape of the scaffold.
Whereas with a baby, somehow the heart just magically self-assembles into the right shape. Can you explain kind of how that does that and why it's difficult to copy that?
So did everybody get the question? So how do we copy development? Or do we copy, is that a paradigm for us to copy development in the lab in the tissue engineering context? The answer is yes, and I'm going to kind of handle that question a little bit later because we do actually show some embryos later in the talk. So if we don't answer your question by then, I'd be really happy to talk further. So yeah, we definitely look to development, embryonic development specifically, for inspiration for some of the biomaterials that we choose, for the growth factors that we choose to deliver, and also what I was getting to here, for the biophysical cues that we choose to deliver. So biophysical cues meaning motion, some mechanical stretch, or electrical signals, or flow of fluids. So these are shear stresses, mechanical stresses, and electrical signals. This is kind of the revolutionary thing that's kind of a more recent development in tissue engineering, is looking just beyond kind of drugs and chemicals and looking to some of these other cues as well. And always looking to nature for inspiration. So nature being either development, wound healing, or homeostasis. And in fact, I'll have examples from each of those three types of systems. So the idea is the biomimetic paradigm, so copying nature, because the better that we do at copying nature, the better the cells will do in the lab. And so in the cardiac context, we can two of the main types of signals that we choose to copy are either the electrical signals that are involved in getting a heart to beat, or the blood flow through the capillary bed, because cardiac cells are really greedy. There's a very dense blood supply in the cardiac tissue, and so we copy that in the lab by outfitting the biomaterials on which we grow the tissues with. We pierce them with lasers so that there's holes through which we can flow the cell culture media. So their food kind of is much more in contact with the cells. And what a lot of my work has been on has actually been on the top here, using electrodes to make mini pacemakers to be able to electrically pace the cells in the lab and get them to beat. And we find that both of these biophysical cues are actually quite powerful in getting the cells to create better tissues. Okay, so this is kind of the, oh yes, question.
What defines better tissues?
Okay, so this is a great question. What defines better tissues? And this is a real challenge especially for someone like myself who's an engineer, coming into a biological context in which we have very different senses of how to measure things. And not only that, very different senses of what poses a statistical difference in a measurement and things like this. And so hopefully, as I give you examples of some of the tissues that we've grown and shown you which ones are better than others, hopefully you'll get a sense of how we do that. But it's kind of a bit of an iterative process. So we see that the tissue is maybe beating a little bit more, and so then we'll decide to make a measurement that's associated with that. Another question?
Is the ultimate objective to replace scar tissue from a heart attack with cells that?
Yeah, so did everyone hear the question? Is the goal of tissue engineering to replace scar tissue? And I would actually say the answer is it depends who you ask. So there is a goal of cardiac tissue engineering to kind of make a Band-Aid for the heart that you can put on the heart after a heart attack to help the heart heal. But the idea is also to avoid scar tissue ever having formed in the first place. And so in that sense, the answer would be no. We're not trying to replace the scar. We're trying to prevent it from ever having formed. But there are people who are particularly studying inflammation response and scar tissue specifically. But this is not our personal goal in the lab. Well, my personal goal. Although colleagues of ours are focusing on that. Was there another question? Okay. So hopefully you have a general sense of what tissue engineering is, and the idea that we're trying to copy nature in the lab. And so now I'm just going to jump into a little bit of cardiac physiology to give you a sense of what are the types of things that we look to in the heart context for inspiration. So the heart, right? This should look familiar to everybody, right? At some point, maybe in the past, some bio class in high school. The heart is a pump in the most simplest terms. It takes blood in. The blue blood is the deoxygenated blood, pumps it out to the lungs where it gets oxygenated, comes back from the lungs and gets pumped out to the rest of the body oxygenated. So basically what the heart does is it distributes oxygenated blood throughout the body. And it needs to be quite strong because, if we look at the schematic of where all of the blood goes in the body, it's really quite remarkable that it's just one piece of tissue about the size of a human fist that accomplishes all of this. Okay, and so this schematic gives a sense of just where in the body that the blood goes, and you've got arteries on this side, the capillary beds, and then veins on the other side. I hope this just gives you an appreciation for the sheer strength of this muscle, okay? And in order to be able to perform this task, given all of the changing, demands of the body, there needs to be, you would imagine there's a control system involved, and indeed there is. So I just want to give you a sense, this is a standard control system thing, diagram where there's, control logic, there's sensors, there's effectors, there's a control variable. All of these things exist in the body. The heart senses what local demands are, adjusts accordingly, and et cetera. Okay, and so the main feedback loops to be able to accomplish this in the body are, not surprisingly, between the brain and the heart, and also the vascular bed. And so there's a feedback loop that involves sensory nerves as well as autonomic innervation from the central nervous system that together accomplish control over the heart rate and vascular tone. So just to say, there are many challenges to the cardiovascular system, okay? Just sitting down right now, not a huge demand on the heart, right? Let's be honest. But if you were to stand up really quickly, your heart rate would really have to change in order to ensure that you wouldn't faint, right? Or if you eat a big meal, your heart rate's going to change, okay? Running for the bus, if you have a fever, your heart rate's going to change, but maybe on the order of minutes to hours as opposed to seconds, needing to respond. So there are different time scales at which the heart needs to respond, in addition to just, needing to respond. So there are other things. If you're training for a marathon like I was, there are much more long-term changes that the heart needs to accomplish. And so, as you might imagine, there are different types of biological processes that are behind all of these control systems. And just as a quick overview, this is a zoomed-in picture of the cardiac ganglion. So these are nerves in the heart that perform some of these short-term changes. Longer-term changes on the order of minutes to days are often accomplished by hormones. And then when you're really talking long-term changes, the heart actually, the muscle itself remodels. So you can actually strengthen the muscle itself, okay? Or it can weaken in response to injury. But we're really going to focus in this talk on kind of the cardiac control on the seconds, kind of the order of seconds. So I just wanted to show you, this is a schematic of the kind of scary looking biological map of cardiac control system. So you've got neurons that come from the central nervous system that go to the heart. You also have nerves that go from the heart back to the nervous system. Okay, so it's a real control loop. But what I really do want, so the simplified version is there's kind of two things that are going on. You're either in kind of two modes. You're either in the rest or digest mode, which is maybe after lunch now, right? So probably most people are in kind of like the upper bucket of rest and digest. Hopefully I'll do a good job of keeping you awake. So this is called the parasympathetic rest or digest kind of autonomic nervous system. When the autonomic nervous system parasympathetic pathway is in control, this is how you feel. You feel like going to sleep. And if the sympathetic nervous system is kind of taking over, this is like the fight or flight response. And so pupils dilate, heart starts to beat a lot faster. There's probably a tiger or maybe the beep of a smartphone or something like this that is making you have a little bit of a panic. Okay, and the idea is that these two nervous systems are kind of in balance. It's not as if one is always on and the other is not, but there's a very careful modulation of the strength of these two nervous systems that work in balance to have very precise control over the heart rate. And what's interesting is that although the heart rate is set by both the rest or digest or the fight or flight pathways of the autonomic nervous system, they have very different dynamics. Okay, so vagal stimulation, which is the parasympathetic pathway, so the pathway that slows down the heart, actually works much faster at getting the heart to change its heart rate. If you were to really stimulate the vagus nerve, you get the heart to stop immediately. So the heart is much better at responding to signals that tell it to slow down than to speed up. So the sympathetic pathway, the fight or flight, is a much different type of time scale. Okay, but very, very exquisite control of the heart rate. We have a question?
Yeah, why does it feel like the opposite?
That's a really interesting question. Why does it feel like the opposite? I don't know. This is something that's always counterintuitive. It was very counterintuitive for me to learn in the bio lab with the rabbits. Why is it so much easier to stop the heart than to get it to start? Do keep in mind though that this is still on the order of seconds. Okay. So if we're looking at sympathetic stimulation, this is still 30 seconds. So what you might feel as something fast is actually probably only going to be 1 or 2, and you could see a big change within that window. But the vagal stimulation, the parasympathetic pathway, is really like on the order of maybe milliseconds. So maybe for the human brain to process it sort of feels the same to us, and so we always think of gas and brakes. Maybe this is just the human mind, the way that we think. But it's often counterintuitive for people to learn that, in fact, the heart can slow down faster than it can speed up. So if there's nothing that you remember after this talk, do remember that, because that's kind of a mind warp. Okay, so I'm telling you that the heart rate can change. I'm telling you that there's a control system for doing so. But does anyone know what the pacemaker is in the heart? Like what these things are actually, which part of the heart tells it to beat? No? Yeah. Pardon? The sinus. Yes. So, yes. Which sinus specifically? We actually have 2 pacemakers in our heart. One is the sinoatrial node, which is what our audience member referred to. So, I actually don't have a laser pointer, but it's this yellow dot right here, the SA node, sinoatrial node. This is the main pacemaker of the heart. This is what receives the input from those neurons. Okay. The secondary pacemaker of the heart is this green dot here, the atrial ventricular node. So this is the node that's between the atria of the heart, which receive blood, and the ventricles, which pump it out to the rest of the body. Okay, you might be wondering, why are there 2 main pacemakers? There's actually a third pacemaker, which is a tertiary pacemaker, which are these Purkinje fibers. So these the fibers that actually we can't see them so well. Oh, here we can. So the fibers that connect the entire ventricle to each other. You're like, why do we have 3 pacemakers? I thought our heart only beats once. But if has anyone ever listened to a heartbeat in a stethoscope? There's like the lub-dub, right? Lub-dub, right? And the lub is are the atria beating, and the dub are the ventricles, because it's kind of a 2-step beat. Okay, and so there's actually a built-in delay between when the atria contract and when the ventricles contract. So this is a really exquisite control system here. So this is the most text-heavy slide that we'll see. Basically, I just want to tell you, so we have these 3 pacemakers within the heart, and they're ruled by what's called overdrive suppression. Okay, so the SA node, which is the main pacemaker of the heart, has the fastest beating rate. Okay, 60 beats per second. Okay, per second. Oh goodness, that would be like a hummingbird or something. Thank you, audience. There's such a wide range of heart rates. Actually, I think the blue whale is the slowest and the hummingbird is the fastest. And I cut and paste this one line in every paper that I write, but I certainly don't hold it up here. So heart rates are actually, there's quite a range in the animal kingdom. Neither here nor there. But the sinoatrial node has the fastest heart rate, okay? So the main pacemaker, the kind of upper management, has the fastest heart rate. And the secondary pacemaker, kind of middle management, is the atrioventricular node, the AV node. So the SA node is always telling the AV node to beat faster than it would normally beat, okay? So in this way, the SA node, even though it's not the only pacemaker in the heart, is the pacemaker of the heart. Does that make sense? Because it's always bossing around the lower levels, okay? And so when we have cardiac pathologies, a lot of times it's because that order's been messed up, because maybe one pacemaker, usually the SA node, is a little bit out of whack, and then everything else follows, okay? The Purkinje fibers also have an intrinsic beating rate, but it's much, much slower. And so really, when we say the pacemaker of the heart, we really only mean the SA node. But there is intrinsic pacemaker activity in other parts of the heart as well. But who's actually doing the beating? Who is the person? Who is the character in the heart that's actually getting it to beat? And in fact, it's these billions of cardiac myocytes. These are muscle cells, and they're really, really amazing. You know, they're the most physically active cell in the human body. They're contracting all the time, billions of beats in a lifetime, billions of these cells. They never pause to rest. And they're just so exquisite. And not only that, they're beating in synchrony your entire lifetime, and they almost never mess up, ever. And when they do, there's usually catastrophe associated with it. So these guys are the guys that are doing all the work. And how do they do that? Question. Yes. Did everybody hear that question? Are these cells the same ones that we're born with? That beat our entire life? The answer is yes. Yeah, we've had these since birth. Yeah, it's pretty amazing. Rats, when they're born with 3/4 of the number of heart cells that they will have for their entire lifetime. So they replicate 1/2 times. But we are born with what we got. So be nice to them. They're pretty expensive. How do they do this? How do they connect to each other so well? So has anyone ever built an electrical circuit before? Ever? Some of you guys? Yeah? All right, so how do you do it? You take wires and you plug wires to other wires, and there are resistors, there are capacitors, there are inductors, there are all these little things, like little bits that you can connect to each other in a breadboard, let's say, and they all have different electrical impedances, right? So the thing about the heart is, for it to be able to beat so well in synchrony, they need to be really well connected to each other. And so what's amazing about the heart cells is that they're very interconnected to each other, not just mechanically, which makes sense because the heart has to beat all at once, but also electrically, because every single beat is an electrical event. And so these the electrical connections between cardiac cells are these gap junctions. So it's actual physical little holes between each cardiac cell that allow ions, which are the electrical charge in our bodies, to flow between them. And so it's a very, very low resistance pathway compared to nerves. It's a very different type of electrical connection than what you would expect, than what you would have in the brain, let's say. Okay, so very, very fast connections and very low resistance. Okay, and very, very dense gap junctions, so that the cells really beat as one giant cell together. So they're mechanically and electrically connected to each other. They're also anisotropically connected to each other. So if you don't know what the word anisotropic means, it means that it's kind of, there are more connections along a fiber than across fibers. And so what this accomplishes is a spatial resistance, so that you have these fibers as opposed to having a big rectangle. Okay, so the electrical signal really moves along a fiber because there's a less resistance down a fiber than across between fibers. Does that make sense? Was there a question? I heard a breathing. Okay.
So, so that it takes some time for the signal to get from the source to the farthest away thing.
Right. So did everybody hear that? So does it take some time? Do signals travel at the speed of light? No. No, they travel at, I don't want to put a number on it, but maybe on the order of meters per second is how fast. And so that's why there's a delay between, some of the delays are actually built into the system between the SA node and the AV node, just allowing for that. But I don't want to put a number on it, I'm not sure, but it's certainly not instantaneous. And in fact, I'll actually show you a map of how fast that takes, and that's why that's why we have an EKG signal, is because that's actually showing the spatial movement of the electrical signal. So this actually answers your question. So how does that signal propagate through the heart? And I don't want to dwell on this for too long, but basically the signal, not surprisingly, starts at the pacemaker, goes to the secondary pacemaker, and then is distributed throughout the heart. And this is in a healthy heart, this is exactly what it looks like, and this is why a healthy EKG is going to look the same for pretty much every individual, because it really does map the electrical signals that would be on the surface of the skin. Okay? So it really travels as a single wavefront. The electrical conduction in the heart, it's the action potential, so the electrical signal at each point in the heart actually has a slightly different shape for reasons that we won't really get into, but they have to do with the ion channels that are in each cell. So each cell type is a little bit different. So the electrical signal at each part of the heart is actually a little bit different. But when you add all those signals up and you also take into account the temporal delay in the signal, this is how you get this characteristic EKG signal that you've probably heard, QRST, so the QRS complex. This is something people often talk about in these in medical dramas. But basically, you're adding up these electrical signal, and they sum into this familiar-looking EKG that you would see on television. So the electrocardiogram has these 5 waves, and since they're so characteristic, we can perform diagnoses on people if that signal is not, if it's perturbed. Okay. So just really quickly, I wanted to go through and show you some arrhythmias. When we have problems in the heart, they're often tied to these conduction abnormalities. So the heart's not conducting the way it should be, meaning that there's something wrong with the ions, that the movement of ions, or maybe there's a problem with the gap junctions between the cells. But with the heart, whenever there's something wrong mechanically, it can almost always be diagnosed electrically. So this is an amazing tool that we have. So this is just an example here of a shortened PR interval, meaning that the delay between the atria and the ventricles is a little bit shorter than it should be. We have what's called a closed conduction loop. This is when gap junctions are kind of messed up, and so you might have resistance that's variable, and so if there's a bit of a block there that's only temporally present, you can get what's called a reentrant loop. And so this is actually a very dangerous situation, because if the heart is conducting signals in a loop, it will beat out of control, and then, as you might imagine, blood is not efficiently pumped through the body. And so this is often something that happens in the atria. I think this is my next slide. When we have atrial fibrillation, this is something that's caused by these reentrant loops. And so what the doctors will often do is find that small piece of tissue that's creating this reentrant loop and actually just go in there and ablate it. So go in there and kill it. Basically, this is the cure. And so really thinking ahead, maybe instead of thinking about how to cure the heart by killing pieces of it, maybe we can think about curing the heart by growing pieces of it or curing it, right? So atrial fibrillation is actually quite an epidemic right now with no real cure. And it's only getting worse. So right now, the only cures are either ablation or just giving patients antithrombic medication, so things to prevent them from having clots, because the real risk of having atrial fibrillation is stroke.
So,
Pardon, question?
Why is it getting worse?
Getting worse because our population's aging, and actually it's a big mystery. So the question is, why is atrial fibrillation such an epidemic? This is actually something that people don't know. Is it some sort of symptom of parasympathetic or sympathetic an imbalance there, where maybe people have too much adrenal fatigue? It's kind of a big mystery right now in that space. Not my expertise, but at conferences it comes up a lot. So VFib, ventricular fibrillation. This usually the next step after this. So you notice it's kind of crazy tachycardia fibrillation. The next step after this really is just a flat line. This is very dangerous. This is usually if this is what someone's EKG looks like, it's probably not going to be an EKG for much longer. So this is when they say, oh, we're in VTach, oh, we're in VFib, and they bring out the defibrillators and the paddles and everything. That's this. This is when the ventricles are going crazy.
So what do the paddles do? How does that fix?
So the question is, how does defibrillation work? And the answer is mystery. Isn't that funny? So some people might disagree with me, but my short answer is unknown. The longer short answer is that defibrillation depolarizes the entire heart. So basically gets every single cell in the heart to electrically get excited at the same time. But for mysterious reasons, that can often reset the heart to beat normally. And that's the mystery part. So we know that the defibrillation actually gets the entire heart to beat in synchrony, but then for some reason it can often sort of pick up where you leave off and be okay. Has anyone ever had a defibrillator used on them? Wow.
Great fun.
He said great fun. Okay. Wow. Compared to AFib. I've heard it feels like being kicked by a big animal. I don't know if that's how you would describe it, but this is how a patient that I met had described it. A lot of people get defibrillators implanted. Okay, so heart attack, again, has a very characteristic EKG associated with it, which I don't really want to get into, but I do want to say that one thing I do want to focus on this slide is that when someone has myocardial infarction, this is heart attack. This is when a coronary artery is occluded, so blocked off, so a piece of the heart is not getting oxygen, and then the heart cells are very greedy. I mean, as you might imagine, it takes a lot of energy for them to beat billions of times in a lifetime, right? So if they don't have oxygen all the time, they're kind of screwed. So they die really fast. So anytime that the heart is exposed to a lack of oxygen, those cells are compromised almost immediately. And because the heart has a really limited ability to regenerate, this is a huge problem and often leads to kind of a spiral in heart disease. So this is all of this to say that this is our motivation. So I'm almost going to start here again and say this is the motivation for cardiac tissue engineering, is to kind of make a patch for the heart, make a patch for the piece of heart that's been compromised by myocardial infarction. Okay, and what we're going to do is we're going to again talk about this biomimetic paradigm and try and see, well, what can we look to in nature to inspire us to be able to grow this heart in a good way? So looking back on all those things that we've gone through up until this point, how can we use that knowledge to be able to grow a piece of tissue? Okay. And so one of you asked a little bit earlier about cardiac development. So one of the inspirations that we always look to is like, well, okay, how do we heal the heart? Well, how did the heart grow in the first place? Maybe we can get some inspiration from there. And actually, what's interesting is that the heart is one of the first pieces of the embryo to manifest itself. Okay, so you have this tiny embryo, and cells are starting to beat. Actually, and even before that, which is what we're looking at here, there are electrical gradients within the developing embryo that recruit cells to grow, recruit cells to what's called the cardiac primitive streak within the embryo, and then those cells differentiate and eventually become the heart. And so is this something that we can use as an inspiration for growing heart in the lab? It has been for some of our studies, which I'll show you. Another type of inspiration, also from the embryo, is electrical currents that have been measured that are more pulsatile in nature. Okay, so the one that I showed you just a moment ago is more like a direct current kind of stimulation, so the kind of current that your smartphone needs to charge up, as opposed to what you get out of the wall, which is AC or pulsatile in nature, right? So these pulsatile signals come a little bit later in the developing embryo. They come when these cells are already starting to beat and they're starting to connect to each other. Okay, so for example, these are two aggregates of cells, embryonic cells, and this was a study I think from the 70s. So really, really good solid work from years ago, showing that these two signals that are shown up here are the electrical signals from one of the aggregates versus the other. And eventually, sitting next to each other, they start to beat together. Do you see that? Yeah, they're kind of beating apart from each other, and then eventually they're beating together. So these pulsatile signals have been implicated in how cells learn to build those gap junctions to each other, how they learn to talk to each other. Okay, this is a little bit further along in development. When we move on into homeostasis and injury, we have those pulsatile signals that are associated with all those pacemakers, right? We have the pulsatile signals that are associated with all of those every time the heart beats, so that blip of the EKG, right? But what we also have, if those cells are ischemic after myocardial infarction, for example. So everybody knows that your body is filled with salt water, right? Salt water that's pretty close to what's in the ocean. So what I often ask my students is, what conducts electricity? People say, electrons, right? And say, well, would you ever want to jump in a pool? Well, why don't you want the hairdryer? No, that's not how you act. You don't want lightning to strike the ocean while you're in it, right? And too close to where the lightning strikes. But the question is, does water conduct electricity? Who thinks water conducts electricity? Oh, no hands going up. Hands going up, right? And electrons obviously conduct electricity, but what is it about salt water that makes it conduct electricity? Right? You got free ions. And that's what we have in our body. And every single cell in your body is filled with ions because we're filled with salt water. What's interesting is that the salt water that's inside your cells is completely different concentrations of ions than what's outside the cells. So anyone know how lethal injection works? No. Potassium. What? So that alters the osmolality of the cells. That is, I'm not sure. So I'm gonna deflect and actually I don't think so. I don't think it changes the osmolality of the cells, but it's definitely a different concentration of potassium than what the cells want. Cells are really thirsty for potassium. They hoard it like crazy, and there's barely any potassium in the extracellular space. And this is how every cell maintains a voltage across it. Our bodies use extraordinary percentages of energy to just maintain these voltages. So they're very important. And so lethal injection works by throwing a bunch of potassium outside the cells and basically stops the heart because the voltage of the heart cells is gone, and so the heart can't beat anymore. Maybe short circuit. Yep. So basically all this to say, if you've got a cell that's compromised, a cell that's injured, it's probably going to be leaking ions. Okay, and so when you have leaking ions, that's current, right? So this is what we're going to be looking for inspiration from when we're talking about injury. All right. So to give you a sense here of what we're doing in cardiac tissue engineering, we're looking for nature for inspiration, right? And in different circumstances, we're going to be looking at different kinds of signals. Okay, so there is a huge amount of work that needs to be done in order to be able to really exhaustively study all this stuff. So I'll give you a short sampling of some of the work that, a biased sample of studies that have been from my Ph.D. and current work. And so giving you an example first, so our research design basically is that we take different signals, and each signal is going to imply that there's going to be a different system that we have to build. So there's actually quite a lot of work that goes into this. And then we have to be able to perform analytics. One of your questions earlier was, how do you know if something's better than something else? Okay, so all of these are very interplay with each other because some analytics that you need to perform imply that you need a visual line of sight to the tissue. Sometimes this isn't possible when we're growing them, and there's a lot of trade-offs, engineering trade-offs when we're performing this research that make it fun or frustrating depending on your perspective. So the electrochemistry of electrons versus ions comes into play quite a lot. If we have short signals, we tend to ignore them. If we have long signals being anything probably longer than about 5 milliseconds, you need to perform some work to be able to protect the cells from any corrosion or any of the chemical reactions that take place in order to be able to perform that transduction. So ideal electrodes need to be biocompatible, injecting charge efficiently, and of course not corroding, because then there's evil stuff in the body or exposed to the cells. Okay, so our example from early development, we have a bioreactor. That's what this is called, a little cell culture system that's outfitted with electrodes. And this is a kind of small system, 1 millimeter by. So the embroid bodies that we're growing are about 1 millimeter in size. So we grow a lot of them together. And what we were trying to study was whether or not this performing an electrical signal, a lot like what you would find in development, would help the cells form better cardiac tissue. And your question was, how do you know it's better? Well, we chose to do a couple of assays being analysis. One was we measured fluorescence of reactive oxygen species, which is a measurement of how well we transduced the electrical signal. And in this case, the measurement was the cells were glowing. Okay, so this was the way that so there was a fluorescent dye associated with this expression of reactive oxygen species. So every time we perform a measurement, there has to be some sort of dye, there has to be some sort of way to visualize it, right? And in this case, we visualized it with a fluorescent dye. So it was kind of cool. We were like in the dark at the microscope performing these like fluorescent, taking all these pictures. We felt like darkroom photographers. A lot of fun, a lot of work. The other measurement that we did was we did another. But these fluorescent dyes all come from. Does anyone know where they come from? There's Nobel Prizes associated with this one. Huh?
Lightning bugs.
Lightning bugs? No. Yes, some of them actually lightning bugs, and some of them jellyfish. They're animals that create light, right? And these proteins, if they're conjugated with other molecules, you can use them to make other proteins glow. So this is a protein for cardiac troponin. So it's a very important cardiac protein, and we were able to see it because it was conjugated to, I don't know whether that fluorophore came from jellyfish or lightning bug. But it's really cool because they glow in the dark. Really amazing. So it's a lot of fun to take these pictures, and what I would love to see one day is like a coffee table book of all these beautiful abstract images that us scientists locked up in the dark by ourselves and have no one to show them to. Okay, so an example of pulsatile stimulation with embryonic stem cells. I did a lot of work on this. This is kind of the bulk of my Ph.D. work. So these pieces of tissues are about the size of a mini marshmallow. So just showing the ruler there. Lots of computer-controlled systems to be able to perform the electrical stimulus, right? And so let me actually just show you. We did a lot of boring work with comparing different kinds of electrodes. I mean, this was like years of my life here. So compared lots and lots of different electrodes and saw how much current went in and, then picked one and then used it and then did all kinds of other things where we did lots of modeling. And we said, okay, well, what's the electric field going to look like where we're growing these cells? This is all like the boring stuff, right? Because what you really want to do is you want to see how well does it work, right? So this is your question. How do you know you did a good job? All right, so let me show you a beating piece of cardiac tissue that was grown without any electrical stimulation, right? And there's like a tiny, you see that up in the upper left-hand corner? Kind of twitching a little bit. You see that? You're like squinting. It is beating. It is beating, but it's not like super dramatic. They do beat, even with no electrical signal. But when you give them an electrical signal, they're like, happy, happy, joy, joy. They're so cute. I mean, I think they're so cute. But to answer your question, which one would you say is better? Right? And so this is the iterative process in the scientific world. How do you know you did a better job? Well, you say, look, I know that that's beating more. So then I now have to come up with a measurement that shows it's beating more. And hopefully, when I write it in my paper, there's not going to be some peer reviewer who says, I don't really like that method. And then it turns into like a peer review kind of thing, right? So sometimes if there's a new method, it turns into a peer review thing where you kind of have to convince someone that the way you're measuring something is correct. If it's a method that's kind of accepted, then you use that accepted method. Okay, so this is kind of like the scientific process. So you ask you're asking all the hard questions, guys, like these philosophical, how do you know you did a better job? Well, in this case, it's a little obvious, but we actually had to do a lot of MATLAB coding just to be able to say that's better, in a way that satisfied us.
So this is beating the one that you showed, each of these. When you say electrically stimulated, that's grown in an electrically stimulated environment, and then...
Yeah, did everybody hear that question? Like, what do I mean by electrically stimulated? I grew those tissues with electrical stimulation for a week, okay? And then they're beating better. So let them grow with stimulation the whole time. But the beating was without stimulation. And the beating is also under pacing conditions. So I'm even pacing this little guy, and it Draw whatever conclusions you will. But
Is the one on the left being paced also?
yeah, these are both being paced. This is the best this guy can do. But if you grow them with that signal, they do much better. Okay, and so another measurement that we did was, again with MATLAB, again with a lot of this image processing, was to be able to do some strain analysis to be able to say, well, how uniform is that tissue beating? And so, as you can imagine, some reviewers were saying, well, I don't know if I like the way you did that strain analysis. And you'd be like, well, I did it, the pixelage of this picture was, and it's a bit iterative there too. So this was another measurement we did to be able to say, they're beating better, right? It's not easy in science to just say, to have these yes or no questions. Well, it's easier to have a yes/no question than a how much question in biology. So this is a real challenge for an EE type person going into a biomedical context. So a lot of interesting conversations there. Okay, so two more examples. One with adult stem cells. Oh, this is one of my favorite systems because it's a question. Yes. Okay, so if I get the question right, you're asking, do we know if on the individual cell level how well they're connected to each other, or are we just macroscopically measuring the tissue? The answer is we are actually just macroscopically measuring the tissue with this assessment. What we also do, I'm only showing you a sampling of how we draw the conclusions about whether, how we did, but we also have to zoom in on the, not molecular level, but on the cellular level. And we zoom in with transmission electron microscopy. We zoom in using some of those immunofluorescent dyes to really tease out how well are the individual cells doing. Because we do need to know that answer. We need to know not just how well is the tissue doing, but how well are the cells doing. Yeah, so very good question. I left that out of here, but really happy to share some of that data with you if you're interested. So talking about the cellular level, actually, on the micro scale, we can do some of this work too. So using some micro patterning techniques with, in this case, a transparent interdigitated electrode of indium tin oxide. Is anyone familiar with indium tin oxide? No? It's a semiconductor and it's transparent. And so it's really nice because it's compatible with a lot of visualization techniques. And not only that, because it's really small, we can grow really small pieces of tissue. So we can get some answers on the cellular level as opposed to the large tissue level. So for several reasons, it's really nice to use some of these smaller scale systems. So again, lots of modeling, lots of rainbows. The modeling always looks like rainbows because we're using FemLab. Well, what used to be FemLab is now ComSol. But then we also zoom in on the cells and we say, oh goodness, look, when we stimulate these cells electrically in culture, not only do we have kind of more of them, right? The blue dots are the nuclei. We stain them blue. So we have more when we're stimulating them. So for some reason, they're proliferating more. But also, these green dots here are gap junctions, okay? We stain for a gap junctional protein called connexin 43, which is the main cardiac protein. And we see that not only are there more cells, but they're better connected to each other. And so we can start to hypothesize that maybe not just in embryonic development, but also in these in vitro systems, meaning systems in the lab, that maybe we're starting to copy some of the right stuff. We had a question.
We've got about 10 minutes left.
Yeah. Oh, 10 minutes left. Good. And I'm on my last example here. So thank you. So if we're going to copy wound healing, so we went through examples where we're copying early development, where we're copying homeostasis, so we're copying a healthy environment. But now what can we do to maybe piggyback on the body's natural wound healing responses? So for regeneration, right? So if we're going to copy wound healing, maybe we also want to have electrical systems that can copy what that wound current is like, what it's like when a cell is injured and ions spill out. So we also need, at that level, we really need to have a direct current kind of system again. And direct current systems are really difficult to build because they're long duration signals. So anything longer than 2 or 5 milliseconds is long in the body, right? And so what we have to do here is we have to actually find a way to shield the cells, which are grown in that chamber up in the top, from any of the chemical reactions that have to take place in order to generate ions from electrons in that saline solution. So there's a saline solution here, power supply comes in, and that's electrons coming in from the wall, right? They get chemically reacted to these silver chloride electrodes, and any of that, crap that's generated that would kill the cells kind of stays in there. But what we have are salt bridges that allow just the ions to move from there to the cells, so we protect the cells. So this is like a lot of engineering challenges that we have to overcome just in order to be able to ask that biological question. So there's a real need for, I hope I'm giving you a sense that there's a real need for collaboration between engineers and biologists in this space, and chemical engineers as well, because electrochemistry, biology, engineering, fabrication techniques, all of these things come into play, and no one person is going to know how to do any of that stuff. So if you look at the papers that come out in tissue engineering, there are often many people from very diverse backgrounds who collaborate. So it's a really kind of fun interdisciplinary field to be in as well. Hope I haven't scared you. So this was a really fun project that we did with human adipose-derived stem cells. Is everyone aware that you have tons of stem cells in your fat tissue? This is something most people don't know. But we get these cells from, people just line up to donate. They're like, yes, you can give me lipo for science. I'll help cure cancer with lipo, right? So there's like, But, people are really excited to donate, but there's plenty of cells. So your adipose tissue, healthy adipose tissue, has lots of stem cells in it. And these stem cells are pretty powerful. They're pretty close to what you might find in the bone marrow. So what's really nice is by exposing these cells to a current that's really close to what a wound current would be, so 6 volts per centimeter over 4 hours. Do you see how the cells are moving and assembling themselves and lining up? I mean, this is amazing stuff. These signals really, really talk to the cells. And so what's unclear is how that patterning that happens from the cells and how that movement of the cells might correlate to what you might find in the body with wound currents. Okay, so this is an open question. This is an active area of research, but it's a lot of fun to do these experiments because you're really watching the cells move, and they're really cute. Again, so we can assess how much did they move, how long did they get, and so then you start doing some of this image analysis stuff to be able to answer those questions. And what we saw is that not only, in this case, we have fewer gap junctions per cell, because the cells are moving around. They have to disassociate from each other to be able to move. And so then more questions open up. What does that mean that there are fewer gap junctions per cell? And so then the science continues. And we also did a really nice experiment. This is all very preliminary stuff with adipose-derived stem cells from epicardial fat. So there are fat pads around the heart, and there are stem cells within those fat pads. And so what we were hypothesizing is maybe there's a role for those cells in wound healing, as limited as the cardiac wound healing process is. Maybe there's something with those cells that we can do. And what was really interesting is that we saw this same movement and elongation with these human adipose-derived stem cells from this epicardial fat. But this is preliminary stuff published a couple years back. So what are we doing kind of now? What are kind of current developments in this field? One thing that people are doing, well, this is actually another example from us, this is in press right now, is beginning to combine modalities. So I had said much earlier that perfusion, like blood, so giving greedy cells lots of food, is really important for cardiac cells, as is electrical stimulation. So one thing that our group is doing and that other groups are doing as well is beginning to combine some of these modalities together, saying, well, what if we applied electrical signals and perfusion? Would that be better? And what we do find, and what we often find with biology, is that things do not sum linearly, okay? A plus B sometimes equals much bigger than what that sum would be. So we're really excited about some of these results. But then again, you see there's another pretty complicated looking bioreactor that we had to build. So there's all this engineering work that goes into asking that question. And so going forward, what would be really nice, and this is another ongoing project that we have in the lab, is to be able to scale up some of these systems. You guys at Google probably care a lot about scalability. And in the tissue engineering context, it's something on people's minds, but we're often building little prototypes all the time, prototypes. But as we start answering some of these questions, then what's going to start to happen is that we're going to need to scale up, not just to be able to build these things, to say, okay, look, we got some answers. We know how to grow a piece of tissue. Now, how do we grow 10,000 of them? And then The answer is duh, like, I don't know. So we have to be able to standardize these culture systems and be able to fabricate them and be able to do it in a scalable way. So this is an ongoing project that we have right now, and we're excited about some patents that we filed. And then finally, let's just say we actually build that patch. Then you have to think about how you actually put it in the body. And so another area that's going to be opening up soon is going to be answering some of the questions of how do you actually put it into a body? How do you make sure that it beats along with the heart that you've attached it to? And so what might need to happen is implantation along with a pacemaker. But these are questions that are just starting to be addressed now. And then finally, if we have all those questions answered, if we finally scaled everything up, and have really great pieces of heart that we can put in the body, then we've got this other really hard problem ahead of us, which I would be remiss if I didn't bring up, which is the gap between the laboratory and the world. Okay, and right now this is a huge issue, which I know Google is very aware of, is how to get really good ideas out of the lab and into the world. And so I felt like I would be remiss without saying that. But this is something that's on every scientist's mind who's in this field, is how do we make it so that even if we've solved this problem, that it actually gets to help people. Okay? And so with that, I'm going to pause again for questions and say, let's, maybe we will get one step closer with regenerative medicine back to our Prometheus, the Greek god of who's formed human beings. And open it up again. Well, I should just say thank everybody, and my dogs, and New York, and wine, and tea, and all those things that make it possible for scientists to do their work. And finally to say, if you have any more questions, I would be so delighted to answer them. And just thank you so much for your attention. This is a real honor for me.